would later base his testimony when defending Burzynski against the Texas Medical Board. This site visit not only confirmed that anti-neoplastons were curing what was previously considered incurable brain cancer, but it garnered their interest in conducting a confirmatory trial under Division of Cancer Treatment sponsorship at the National Cancer Institute. These trials involved most of their top experts, including Dr. Michael Friedman, the Associate Director of the Cancer Therapy Evaluation Program. In a memo addressed to his director, Dr. Friedman wrote, "I thought you would be interested in this. Anti-neoplastons deserve a closer look. It turns out that the agents are well-defined, pure chemical entities. The human brain tumor responses are real." The National Cancer Institute's decision network then convened and gave the green light to conduct government-sponsored clinical trials of anti-neoplastons.
Initially, everybody was very excited about it. Everybody would like to proceed. The people who reviewed our results, the experts from the NCI, they did a very good job. They were critical, of course, but they were also highly complimentary for the way we treat patients and the results we got. Okay, so it looks like everything should open and we move forward. Suddenly, everything came to the stop, and then we found that a few months later, Elan received permission to do clinical trials with this particular ingredient. Okay, ours was pushed back for something like 4 years, and then Elan was allowed to proceed with this one suddenly.
When Elan terminated their business deal with Burzynski, they went behind his back, recruited Dr. Davorit Samid, and partnered with the National Cancer Institute, where Dr. Samid soon became a section chief. Elan then co-sponsored laboratory research and clinical trials testing only this single ingredient, called phenylacetate, the same chemical that Burzynski was advised he couldn't patent, and had already proven to be quite limited against cancer as a single substance as far back as 1980.
After the treatment in a small number of patients, we found that the activity was quite limited. That's why we decided to abandon phenylacetate and we use the other anti-neoplastons. One of them contained phenylacetate as the second ingredient. Phenylacetate alone has very small activity. It's not very effective.
In 1994, I started working for Davorit Samid. She didn't let me know that one of those compounds is from Burzynski, Dr. Burzynski. You know, it's just say phenylacetate and show me all the published paper about phenylacetate and its analogs and their anti-cancer activity. It was quite amazing in the lab, because if you find a compound that has anti-cancer activity and then you find a bunch of analogs, it's like you stumbled in upon a pile of gold. So people would think, hey, say, you know, patents, you know, that sort of thing. Davorit, under her leadership, we find a lot of biological activities of these compounds. So it would have anti-cancer activity. So the scientists at Johns Hopkins tried to patent these compounds. But of course, Davorit was working with Elan pharmaceutical company at that time. So those guys at Johns Hopkins didn't have any chance of patenting those compounds. So, you know, but it's interesting that she would complain in the lab and saying that these guys, you know, it's try to, you know, go behind her back and patent these compounds.
While Burzynski was facing continuous harassment from state and federal agencies, the earliest phenylacetate studies were published in April of '92, authored by Davorit Samid, hosted by the National Cancer Institute. Burzynski sat in awe as he witnessed the National Cancer Institute recruit one of his researchers, push his research aside, and begin to test phenylacetate without him. reporting phenylacetate is both effective in inducing tumor cell maturation and free of cytotoxic and carcinogenic effects, a combination that warrants attention to its potential use in cancer intervention.
1995, in the lab, I was still with devoid, but I smell fishy. Something is not right. The first paper devoid published about phenylacetate, if you look at the methodology section, there's the BRI abbreviation. She got materials from Burzynski Research Institute, Houston, Texas, but it didn't say Burzynski Research Institute. For us, as scientists, it's a funny practice.
Burzynski's name failed to appear in the acknowledgments or any of the references listed in this report. Burzynski knew these tests would fail, since he had already proven this in his own laboratory 12 years before. Abandoned by the National Cancer Institute, he sat powerless on the sidelines as the attempted hijacking of his discovery unfolded before his eyes, ending in the hideous train wreck he warned them it would. The National Cancer Institute, Elenin, and Dr. Samid spent over 4 years and tens of millions of dollars testing phenylacetate.
Phenylacetate really by itself has very little clinical effect.
And the NCI or Elenin could not use the other ingredients of angioplastin because they were covered by the patents owned by me. They were trying to commercialize this, but without the other ingredients, they couldn't do much with this. This needed to be given in conjunction with the others.
While coming to terms with this reality, the National Cancer Institute decided to honor the government-sponsored clinical trials of anti-neoplastons they had initially promised Burzynski in 1991. From the moment this dialogue was reopened, the National Cancer Institute proposed altering the treatment protocols that Burzynski had spent 20 years perfecting. Burzynski told them that until they agree to a protocol that he has confidence in, he was not going to provide the National Cancer Institute with any anti-neoplastons. Dr. Michael Friedman told Burzynski, in response to your correspondence, one last time, we will revise the protocol with regard to dose and schedule in compliance. However, if you are unable or unwilling to provide the anti-neoplastons in the near future, we will pursue alternative sources to procure the drug or its active components, and will proceed. Burzynski responded, I appreciate very much that you have finally decided to follow my recommendation regarding dosage, but your final statements that you are ready to proceed with the treatment with anti-neoplastons without our participation caught me by surprise. It is hard to imagine that a federal employee would consider patent infringement. Placing these ominous threats from Michael Friedman aside, they finally managed to agree on a protocol. The protocol was simple. Patients with tumors larger than 5 centimeters, more than one tumor, or with metastasis are excluded. After everything was approved and Burzynski released his medicine, a full year passes, in which time over 15,000 Americans had been diagnosed with the types of brain cancer this trial was focusing on. Yet, America's National Cancer Institute was somehow having difficulties in accruing patients. So, they used this as an excuse to go behind Burzynski's back, again, and take it upon themselves to drastically alter the protocols. Dr. Friedman, it has been brought to my attention that the protocol has been amended to accept patients with tumors measuring over 5 centimeters, multiple tumors, or with metastasis. I am outraged that without my knowledge, Memorial Sloan Kettering Cancer Center, with NCI's permission, changed the protocol. Treatment of patients with these conditions will require a different protocol with a different schedule and different dosages. I hereby request that the amendments be canceled immediately and the original protocol be used as promised. The National Cancer Institute responds, sorry, but the amendments have been approved. Burzynski fires back. Let me make perfectly clear that as the discoverer and developer of anti-neoplastons and the individual with nearly 20 years clinical experience using them, it is my professional opinion that the drugs will not produce substantial benefit in such advanced patients. As I have repeatedly informed you, the protocol that we are currently using for such advanced tumors requires doses 3 times greater than that currently being used.
These trials have been conducted at the Mayo Clinic in Minnesota and the Sloan Kettering Hospital in New York. Currently, there are only 8 patients enrolled.
I'm very dissatisfied with that because our desire is to achieve the proper enrollment in as quickly a manner as we can in order to really test this hypothesis.
Friedman blamed Burzynski for this slow motion, saying Burzynski restricted admissions to the trials too stringently. To speed things up, admission standards for the trials have been lowered to bring in patients in worse physical condition. This over Dr. Burzynski's strenuous objections.
We got the idea that the main interest is to let this patient die rapidly and make sure that treatment will never work.
Dr. Burzynski has threatened legal action to halt these new admissions, and NCI has suspended recruiting, leaving the trials with just those scientifically unsatisfactory 8 patients. Meanwhile, both the FDA and the Texas Medical Board are still trying to stop Burzynski.
On May 8th of '95, less than 2 weeks before this broadcast, the National Cancer Institute issued an internal memo to all of those involved in the anti-neoplaston trials. For the record, the Clinical Trials Monitoring Service has been instructed not to send any anti-neoplastons clinical trial data to Dr. Burzynski, the Burzynski Research Institute, or anyone inquiring about the anti-neoplastons clinical trials. Any inquiries that may be related to the trials or Dr. Burzynski are to be referred to the Associate Director, Dr. Michael Friedman. Friedman then taunts Burzynski. I must convey my deep pessimism. We are in no way obligated to obtain your consent. Your insistence on dictating the manner in which we conduct or review these clinical trials is both presumptuous and inappropriate. The future of these trials rests entirely with the NCI, since our primary obligation is to the American public. Burzynski responds, your letter of June 6 conveys pessimism. My letter conveys outrage. Patients were admitted against admission criteria, their treatment was discontinued, and their lives were jeopardized for frivolous reasons. In spite of your promise, we never received any detailed data on these patients. There must be a reason why you are afraid to provide us with complete copies of medical records. Well, there was a very good reason why Dr. Michael Friedman was afraid to provide Burzynski with complete copies of these patients' medical records.
It took us at least half a year before we forced NCI to release some of this information to us. And then we found that they severely violated protocol. They did not comply at all with the protocol. On the top of that, patients were forced to stop the treatment with antioplastons because of massive fluid retention. And this is something which we don't see with antioplastons. Typical effect of antioplastons is dehydration, which means elimination of the fluids. The patients are losing a lot of fluids to the point that they have to drink a lot of extra fluid. We don't see increased fluid retention. I was curious why this could happen. I knew that the patients were receiving a lot of intravenous fluid, but then we learned that perhaps the fluid which they were receiving were not anti-neoplastons.
In October of 1995, the National Cancer Institute's Cancer Information Service issued a public statement for anyone inquiring about their clinical trials of anti-neoplastons. In it, they stated, because these studies were closed prior to completion, no conclusions can be made about the effectiveness or toxicity of anti-neoplastons. To their credit, and according to the scientific standards set by the National Cancer Institute, this was indeed the truth. However, 4 years after these trials were closed, and 2 years after Burzynski defeated the FDA and won his freedom, the National Cancer Institute just couldn't leave well enough alone and decided to vindictively publish these scientifically invalid anti-neoplaston trials in the peer-reviewed medical literature. In it, they described how 9 patients were treated, and no patient demonstrated tumor regression. However, whoever was responsible for publishing this report was apparently careless enough to also include the anti-neoplastons concentrations detected in the blood of the 9 patients during treatment.
We compared this to the data which we have in our studies. We found that they were severely diluting the medicine, and this was why the patient had fluid overload. Antioplaston AS2-1 consists of two ingredients, which is called phenylacetate and phenylacetylglutamine, and about 2.7 times lower level of phenylacetate in patients' blood compared to what we see in our patients who receive successful treatment. Phenylacetylglutamine, there is about 36 times lower level in patients' blood compared to what we see in our patients receiving the right dosage of antineoplastons. And the concentrations of phenylacetyl isoglutamine, one of the main ingredients of antineoplaston A10,